Oh, good. Okay, you guys have to help me because disaster is afoot. Well, we've been all worried about coronavirus. I did something bad. We were trying to study Ebola, obviously, from yesterday. And I'm trying to study these sequences. So I just took out some of the old virus from the freezer. You know, no big deal. Just trying to study, sequence the DNA. But I did an oopsie, and now Ebola is free. So now you have to help me find them. Okay, so let's come with me. Come with me. Let me know if you see it. Hi, Jack. Ah! Oh no! Do you guys see it? It's right here. It's right here. Ebola. Okay, now we're gonna have to do this very carefully, but we're gonna have to extract the Ebola from Jack. He has no idea what's happening. No idea. All right, ready, Jack? 1, 2, 3. Ah! Whew. Okay. That was a close one. Okay. This is the specimen that got out. Okay. It's all right. I sanitized him. So he's done. Sanitize him. It's fine. This is now a dead Ebola. Okay. This is what they actually look like. So we're just going to study the structure because it's dead. No big deal. They are called filoviruses because they have, because they kind of look like worms, and that's how they got their name. So it's just kind of like one big string, and that's part of what you guys are going to do today. You guys are going to, we're going to go over part 1. I'm going to introduce part 2. We're going to do part 2 together of the Ebola disease detectives. And then we will, you guys are going to go on to a virus explorer activity and kind of study Ebola. And then tomorrow we'll talk about the difference between Ebola and coronavirus. All right, so let's get to part 1. Okay, part 1. Let's do it. So you should have watched the video yesterday and answered those 3 questions. The 3 questions we're talking about those scientists that were studying the Ebola outbreak and 3 factors that really caused that outbreak to occur. So one was the high density of bats in the area because it's a zoonotic disease. We call it zoonotic, meaning it can pass from animals to humans, and that's how it was originally started. It was in bats originally and then passed to humans. But why this outbreak took hold as opposed to just dying in one human and then that being it is that there was a high density of bats and a high density of human population. This occurred in Africa. In Africa, they have a poor healthcare infrastructure, so that did not help the situation. And infections began to occur in healthcare workers themselves, meaning they were passing them to their patients without knowing they were infected originally, or patients were sick and they were getting it, and then they were getting it sick and dying. So that's why it was so bad. But also you had the virus itself that was mutating to become more spreadable, more transmissible. So we're going to break out the whiteboard real quick. If you're taking my disease class next year, this is just kind of an introduction of one of the things we're going to be talking about. But let's talk about what the virus's goals are. Like any virus, what's a goal for it to have? Well, it's the same as any other organism, which is to have more reproductive opportunities to pass on its DNA. So pass on its genetic information. So the more it reproduces, the more successful it is. So its goal is to reproduce and pass on genetic information, just like any other organism. And there are two big factors that allow for this. Transmission is the main one. This is how quickly something spreads. Transmission is the ability to jump from one organism to another. So the higher transmission you have, the more reproductive opportunities you have. We use this in the field of disease, what we call disease ecology or epidemiology, is this term R naught, which is like R with a little subscript of 0. R naught actually measures transmission. So you may see, we're not really going to talk much about this, but you'll see this in turn. If you're reading an article about coronavirus, R naught will likely come up because coronavirus has a high R naught. And virulence is the ability to cause damage or disease. So this term virulence means how damaging it can be to an organism. And usually there's different ways to measure virulence, but sometimes we'll use death rate as a way of measuring that because the more disease and damage it causes, the higher virulence it has, and you can't have a higher damage than death. So the death rate is can be used as a measure of virulence. Now, we had our original, you should have cut these guys out. We had our original reference DNA, okay? This was taken from an original case in Guinea, okay? So the Ebola virus we had sequenced before the outbreak, before this current outbreak occurred, was this one. And this one's from the 70s and in an outbreak that occurred a long time ago in Guinea. So this is like our OG, okay, our reference. The cat's currently trying to play with Ebola. Are you playing with Ebola? Oh. The cat has eradicated Ebola. Anyways. Okay. Anyways, so if something has a higher virulence, usually means it has a lower transmission. Because if it kills its host faster, so Ebola in this original sequence was super deadly. It was like 60 to 90% death rate, which is huge. So you're pretty much screwed if you're infected with it. In this original sequence from the 70s, it had a really high virulence, so it's really high death rate, which meant its transmission was lower. Because if it caused death so quickly, then it's less time to actually transmit to another organism. Or if someone looks like they're really sick and about to die, no one wants to come near them because they know they're sick. So really, this one was not as successful, this reference. However, what happened is, going back to our sequences. Okay, this is for part 2, how they wanted you to arrange your sequences. Okay, so we have our reference, our original guy up here, and this is how they wanted you to lay those mutations out for part 2. Basically what we're going to go through is that there were mutations that occurred in these sequences that made this Ebola outbreak more successful than this original one. So we're going to look into that. So looking at to finish off our part 2 and 3 here, on our first page, it wants you to define term mutation. You guys know what a mutation is. It's a change in the DNA sequence that has not changed. That's still the definition of a mutation. And 3, why is it important to examine the sequence of Ebola during an outbreak? So why would we want to take these sequences? These sequences are actually from different patients. So during this current Ebola outbreak, or the most recent Ebola outbreak, these were taken from samples of blood from different patients. And we were actually able to get the Ebola virus from each of these different patients. So we have 1 through 15 15 total patients that we got Ebola out of. Why would we want to do that? Well, you want to know if Ebola is accumulating mutations that help it to spread. So epidemiologists want to know if it's mutating to spread. And we would want to know that for coronavirus too. We'd want to know if it was mutating. And if you have a DNA sequence, that can sometimes help you to diagnose if someone is sick or not. Some tests actually test for DNA of a virus to see if you were ever infected. That's what we used to do in the lab when we had amphibians infected with a virus. We would basically dissect or cut part of their tail off. They regenerate their tail, so it's not an issue. But if they came positive for ranavirus, the virus that we were studying, we know that they were they are infected with it, or they were at some point infected with it and have recovered. So that's one way you can kind of test to see how that virus has spread through a population. So that would be number 3. In part 1, what it had you do is basically group these guys. Group these guys in ways that you thought they should be grouped. So similarities and differences. Honestly, in part 1, they're just trying to get you to look at patterns, to see patterns between the sequences before we go to part 2, where it's actually showing you what the best sequence, what the best groupings are. And we're going to go through why that is. But part 1, number 1, you're all going to have various different answers for how you decided to group them. But you probably group them by similar mutations or similarities in the sequence. And number 3 is an important question. It asks if a sequence has a larger number of mutations when compared to this original reference sequence. What does that mean? Does that mean it's from earlier or later in the outbreak? Well, the more mutations it accumulates, that means it had more chance to replicate and pass on those mutations. So the more mutations, the more it's replicated, that means it's later in the outbreak. Whereas if it only has 1 or 2 mutations from the original sequence, then that's earlier in the outbreak because it hasn't had as many opportunities to reproduce. So less mutations is earlier in the outbreak, and more mutations is later in the outbreak. All right, so now going to part 2. This is how you can arrange these. You can pause and arrange them how I have, just so you can kind of see. But it's having you, I just followed the same sequence that they had here. Okay, so it's showing the reference. This is increasing through time. All right, so we have our reference. Then we can see we have a change from our reference sequence to this first grouping of sequences here, group 1. And the big change is we have 2 key mutations here in group 1. We have C and T. So originally in our reference, this was a T in nucleotide 2, but it has changed as mutated to a C. And then here in nucleotide 6, we has gone from a C and mutated to a T. Okay, so we have 2 core mutations there. So these are likely early in the outbreak because they only have 2 differences from the original sequence. There's Pepper, in case you want to know who she was. Pepper, say hi. Oh, she looked at you. That's the most you're going to get. All right, then you have group 2. So we have group 2 here, which is sequence 2, 5, 7, 11, 10, and 15. And they have the same sequence differences. So if you line them up with group 1, you can see they also have that C. They retained that original mutation, but and the nucleotide 6 is T. But in group 2, we have even more mutations that accumulated. We actually have 4 more. Woo. So we also have in this group, making sure I have it right. Oops, this guy belongs up here. So that didn't look right. 2, 5, 7, 10, 15. My bad. Okay, so there we have these mutations, these 2 core mutations, as well as we have nucleotide 1 has a new mutation. We have nucleotide 7 has a new mutation. And then we have one in 12 and 13 also. So we have 4 more mutations in addition to the original 2 that were up here, meaning that in these patients, these are patients that were later in the outbreak because they have more mutations from the original sequence. This virus is now, with these mutations, getting better at spreading, better at the transmission part. And then our last 2 patients, we have all of those previous mutations. So you can kind of compare this to all the previous groupings in number, you guys can't see that, in number 8 and 12. But we also have, in addition, we have that very last nucleotide that's accumulated. So from the original, this one has 7 total mutation differences. These 2 patients do. So these were the last 2 patients we sequenced in the outbreak. They are most different from our original sequence. So they must have been later in the outbreak. Okay? And this is true for how natural selection works too. You can kind of tell how far something is related to something else by sequencing it and seeing how many differences there are. So this concept is applied not only to disease outbreaks, but it's also applied to figuring out like how 2 species are related to each other. Okay, so you have this reference. This guy, remember, he was really deadly, had high virulence and low, kind of less transmissibility, less ability to spread because he was so deadly. And now we have, so that was this guy, lower transmission, higher virulence. But now we have later in the outbreak, our 8 and 12, where we have 7 total mutation differences, we're seeing that's becoming more transmissible. So it's higher transmittance. It's able to spread more easily with those mutations. And as part of that, what actually happened in this Ebola outbreak was, interestingly enough, it actually had a lower death rate over time. So these mutations that it got basically had an effect on virulence, made it less deadly, but made it more successful in transmitting itself, more successful at passing on its genetic information. So that's what these mutations often do, is they help the virus to spread so that it can pass on its information. So our later viruses were more successful in terms of the goals of a virus. Now let's go to the part 2 questions. So we have number 2, A, difference between the reference sample and group 1 were those 2 core mutations. Differences between groups 1 and 2, we had those original 2 plus 4 more. So we had 6 total mutations from the reference, but 4 differences between groups 1 and 2. And differences between groups 2 and 3, we had 6 of those original mutations, but we added 1 more core mutation from that. I wrote the wrong number. So we had 1 mutation difference between group 1, 2, and 3. Okay, so that was part 2 together. So we kind of answered some of these already, but let's go through some of them together. What can you infer from this diagram about when each group of patients contracted Ebola relative to the outbreak? Well, we have the ones with less mutations, the ones who had a virus that had less mutations were earlier in the outbreak, and the with more mutations occurred later in the outbreak. Explain how the sequences and groupings support the hypothesis that mutations accumulate over time. Each one of those had these, because they each had the original mutations plus more, that means that they were accumulating instead of becoming different mutations. It's more logical that it's that original virus, the progeny of our reference, that's actually just accumulating more mutations as those offspring are allowing more opportunities to replicate. So it's accumulating more mutations over time instead of getting new mutations. Because of those, it had all the original mutations plus more. how can you explain the fact that some sequences have an additional non-core mutations that did not spread to other groups? So for example, let's take sequence 3. Sequence 3 was in group 1. And in group 1, we had this mutation here in nucleotide 2. And we had this mutation, you can kind of see that shaded. There we go. This mutation here. All of the ones in group 1 had that mutation. But this one also has a G. It has one more, this sequence has one more mutation. But why didn't this G mutation take hold? Why isn't this mutation passed on to the next group later in the outbreak? Well, likely if this mutation didn't take hold, then it didn't really make much of a difference in terms of transmission. So it either didn't give the virus an advantage at all, or it would cause a disadvantage. Remember, not all mutations are bad and not all mutations are good. Some of them can result in nothing. So that might be this in this case. If it doesn't give an advantage, it's likely not getting passed on to the next group, to its progeny. And number 6, if a particular mutation was advantageous to the virus and that it allows the virus to spread faster, what would you notice happening over time to all the sequences you collected? Well, that's what occurred here. We had, for example, in this group 1, we had this C and T mutated here. And that was passed on to all the progeny after that. So all the successful viruses that are able to cause disease in another organism will include that mutation if it's advantageous. Okay, so it's just like getting an additional trait that's helping it to reproduce. If it's good and it helps to reproduce, then it's going to be passed on. And that's the end. Okay, so that would be part 2. We've done it together in its entirety. Now what you guys are going to do today, this is going to be pretty short, honestly. You're going to go to this Virus Explorer right here, and you're going to check out Ebola virus. And you're just going to see, like, look at its structure. It has you figure out is it enveloped? Does it have, what shape is it? Genome type. So you're just playing around with that. And it's honestly a cool thing just to look at. It compares a lot of different human viruses, as well as some other common viruses of other animals and plants. So go ahead and check that out. That's your assignment today, is just that extension activity. And it is pretty short. And we will be back tomorrow. Remember, you do have a quiz. It's open note over this stuff. It will be a timed thing, so it's not going to be a Google form. It's going to be a Flexi quiz, I think is what the app is called, but you're going to have a link to it that you just go to. But once you click on that link, it will be a timed 30-minute quiz. It's not going to be that long, but you should be able to finish it in 30 minutes, no problem. If that is an issue for you, just contact me. I might even make it 40 minutes. It's just so you guys, to discourage you guys actually communicating between each other. That's what it's for, just so you know. Okay, so have a great day. Get out in the sunshine. Bye.